And we're live. Okay, so we're just going to hang out here for a bit, waiting for people to join. I guess I should have asked for a critical mass or something, a number of people to see join, but I'll survive. Actually, I'm realizing that I can't see it on the other screen, or can I? Where do I see how many people have joined? Hey guys, sorry to throw you under the bus, but quick technical question. Where do I see where people have joined?
Can you hear me? Oh yeah, on YouTube, directly on YouTube.
Oh,
you can also see at the top of your screen when you're live, you've got a little eyeball with number of viewers there.
Thanks. Sorry everyone. Sorry guys.
No worries.
All right, we're at 11 people so far. Just want to give it a sec. I think we have over 40, maybe 50 people signed up. And I don't want to kick this off too early. Thank you. 21 people. I think I should go now. 24 people. Apologies to the people who got here exactly on time and have to watch us just kind of twiddle our thumbs for a second, but the second slide is real good and we don't want people to miss it.
Maybe tell a joke while we're waiting.
Maybe I'll show off my mask. It's got sharks. We should have done this whole thing with masks on just to be extra safe. Okay, I'm going to kick this off. Hi, and welcome to DEPT Talks Live. Today, we're going to talk about some work that we at Rocket Insights are doing with Goldfinch Bio, one of our clients. We're helping their scientists develop new treatments for kidney disease by building software. So this is a quick overview of sort of what we'll be talking about today in order. Just a little about us up front, talk about background on kidney disease, the Kidney Genome Atlas, which is the software that we've written and we're going to be presenting. We're going to talk a bit about the implementation of that, not just the engineering, but sort of big picture. And then big picture, talk about the future, not only for Goldfinch, but also just in biotech. So, us. Conveniently, we're both named Adam. I'm Adam Fraser. I'm a software engineer at Rocket Insights. DEPT is our parent company and the host of these talks, so thank you. They do all the things digital, everything digital, basically, but we at Rocket are just focused solely on product design and engineering. So we do web, iOS, and Android. I've personally been an engineer for a little over a decade and have a mixed background in design and visualization, and I've worked for some biotech companies in the past. And Adam?
Yeah, hi everyone. I'm Adam Tebbe, Senior Director of Informatics and IT at Goldfinch Bio. I've been at Goldfinch for about 3 years now in this role, helping to build our Kidney Genome Atlas. Goldfinch has only been a company for about 3.5 years now, so it's still very early in our journey. We're a company solely focused on precision therapies to treat kidney diseases. We have multiple programs in various phases of development to address a huge unmet medical need.
All right, big picture. How many people are affected by kidney disease?
Well, chronic kidney disease, or CKD, causes more deaths than breast cancer or prostate cancer. It is the under-recognized public health crisis. It affects an estimated 37 million people in the US, or more than 1 in 7 adults. 1 in 3 American adults is at risk for CKD. CKD is the 9th leading cause of death in the US. Only 23,400 Americans received a kidney transplant in 2019, while more than 100,000 Americans are waiting for one. Less than 1/3 of these transplants came from living donors. Living and deceased kidney donors are crucial. 12 people die every day while waiting for a kidney transplant. Dialysis and transplant are very much still the standard of patient care for most patients. New treatments are desperately needed to help this underserved patient population.
So treatments are needed. What was the impetus for building the KGA, the software, in the first place?
Over the past several decades, many companies have tried to develop new therapies to treat kidney disease with few successes on the market. Kidney disease and its various forms is a polygenic disease that is not very well characterized genetically. Goldfinch's approach has always been that we're not going to develop a drug to treat every single person with kidney disease, but rather take a personalized medicine approach. Therefore, the approach Goldfinch has taken to build its drug discovery pipeline is to start with patients with kidney disease. It's been shown that drug targets with genetic evidence have a twofold or higher likelihood of succeeding in phase 2 and phase 3 trials. Goldfinch has been collecting samples from affected patients, sequencing them, and enrolling them into our Kidney Genome Atlas to try to build a data set large enough that we can use statistical methods to identify potential drug targets with genetic evidence.
So what led you to hire Ring and Rocket Insights to do this in the first place?
Yeah, we were fortunate enough to launch a partnership with Gilead Sciences last year to expand the scope of the KGA to include diabetic kidney disease. Roughly 40% of all diabetics develop kidney disease, and even higher in some racial and ethnic groups. The strategic collaboration aims to discover, develop, and commercialize a pipeline of innovative therapeutics for diabetic kidney disease and certain orphan kidney diseases. Goldfinch is a very small company with limited engineering staff, so we decided right away that we needed external help and staff augmentation. We had a strong desire to build a dynamic web-based application where we could surface all of the data we were collecting and producing as part of our Kidney Genome Atlas to help optimize target identification, patient stratification, and the exploration of the relationship between genotype and phenotype. We set a pretty aggressive timeline to launch the MVP and spoke to a few companies before coming to Rocket. One of our employees had previous experience working with Rocket, and we were really impressed by the breadth and depth of capabilities. They had no concerns about delivering on our timeline. We started with a design sprint and moved quickly into development. Not only did we produce the MVP on time and on budget, but have continued the project, adding a tremendous set of features and capabilities to the application.
Well, let's show them off. So let's talk, actually, before we show them off entirely, what exactly is it? What is the Kidney Genome Atlas? Let's talk about that.
So there's two main components to Goldfinch's platform for target identification and validation. The first is the Kidney Genome Atlas, or KGA for short, and the second is the Human Biology Platform. The KGA is a patient registry and analytic platform designed to store, process, analyze, and display multi-omic data on patients with kidney disease. We partner with hospitals, academic researchers, biobanks, and public consortia to acquire samples and data from patients with various forms of kidney disease. Our first study included in the KGA is centered around FSGS, which is a rare form of kidney disease. In this study, we sequenced about 1,500 patients and more than 1,700 controls, and then added to this an additional 20,000 controls from public repositories. All of this data was whole genome sequencing, with some patients also contributing RNA.
It's a lot of genomes, not to mention clinical data.
Yeah, our first study produced more than 2.5 petabytes of data. In addition, we have numerous automated pipelines for aligning the various forms of sequencing data, doing QC, and then downstream analysis. There's just a lot of data being produced that needs to be organized, collated, and presented in a coherent way. This is what really drove the project with Rocket. We wanted to make the data available to anyone in the company who could use it and not have it locked up in a silo with the computational team who produced it. We decided early on in the development of the KGA that we needed to democratize the data so that we could make informed, data-driven decisions for target discovery and patient stratification. While there are public portals that surface some of the types of data we're producing, we're a commercial organization and not able to share our data in the public domain. We evaluated and even used some commercial products in the past, but they just didn't really meet our needs, and decided ultimately that we needed to build something custom to support our organization.
Just a quick aside. So I remember when we first talked about doing this talk, it was sort of a general audience thing. And I realized we use the term petabytes here. And I feel like probably a general audience would not even have heard of that term before. It's certainly when I heard it, I was like, I don't know that I want to work on this project. It sounds insane, but it's been fun. So one last thing before we get into sort of demoing this, can you talk a little bit about who is using this?
Sure. The current set of users includes a cross-functional team that's really focused on target discovery and validation. So this includes biology, drug discovery, translational sciences, clinical sciences. We're also currently expanding the reach beyond this subset of users to all of those departments, which effectively is our R&D organization. As I said, we're a small company. Requirements are primarily provided by our statistical genetics and computational biology team to make sure that we're accurately and succinctly presenting all of our genetic analyses. Currently, the application is heavily focused on genetics, but we're also working on incorporating several public data sets and features to further enable our drug discovery and biology teams.
All right, so let's get into it. The KGA application is broken up into a few main sections. And at this point, we're sort of just going to walk through what those main sections look like and talk about some of the features that you're seeing here in these animations. So the first section you can see selected in the top tab there is studies. So users upload studies, and studies can be analyses that are done using either public or private data. We currently support GWAS, differential gene expression, and RVAS studies, or rare variant analyses. And each of those has its own sort of interactive data viewer, like what you're seeing here. So what we're looking at in this particular view is 3 GWAS studies that have been loaded. We have stacked them vertically so you can sort of compare where the peaks are. And at the very top, there's a consensus where we report, in this case, we're reporting SNPs that have been hits in all of the studies above the selected threshold. There's a lot of features in this thing, and hopefully we'll hit on some of them in the later slides. But if people have questions sort of at the end of this, we're going to pause before we get into the rest of the presentation if people have questions about anything specific they see here. So another view of this, did I go to the next slide? Let's see. I believe this is a target creation. So here we're clicking on a target track above and then identifying a locus of interest. So this is especially useful for the target discovery group, or target discovery and validation, I think they go by. So the idea is that we have little algorithm on the client side that does peak detection and finds where all these points lie, orders them, and allows them to sort of jump through them quickly. So as new studies come in, they can identify new potential targets and then track them in this dashboard, which we'll talk about in a bit. Let's see, did I miss anything here? So we have gene tracks displayed. You can see those at the top of the Manhattan plot as well, and some other stuff that I'm going to talk about in a minute. I don't know if you had anything to add, Adam.
No, I think it's really helpful to have how you have the arrows as well, so you can jump from peak to peak. So you're not zooming out and zooming back in constantly on the Manhattan plot, but you can just jump from peak to peak really quickly.
Yeah, and this current implementation, they're sort of sorted by like the most important peak first. So ideally, at some point, you go, you know what, I don't really care about the rest of the peaks because most of these are less interesting data, because some of these studies, at least the ones that aren't underpowered, have quite a few. All right, so this one, I put censored on it. That's for the lawyers. So here we're looking at differential gene expression study. This is private data, so you can imagine, you know, sort of whatever you want below. But what we would see below is the same thing as what we had in the Manhattan plot, where we have tables that show the points that are currently in the view. You can zoom in and out on this. Each of these points is a gene, and you can see whether they're positively or negatively expressed and what the sort of level of confidence is for that on the vertical axis. So a volcano plot, I mean, again, it's another type of scatter plot. It's just that something that turns out biologists or geneticists or the scientists that are using this are comfortable with using. So if you're in the sort of data side, you might not have heard of this term before. So hopefully that's helpful. But there's also brushing and picking. So if you were to like hover over points on the table or in the graph, it sort of shows you where it's highlighted in the other place and tells you information about it. Anything I missed here?
Yeah, I think just how you've, you know, on any of these study viewers, we've tried to integrate a search box so it's very quick for someone to jump to a particular gene or variant or locus of interest, depending on what study they're looking at, and just makes it quick to kind of look at your favorite topic of the day and what you're trying to investigate.
Yeah, actually, and this that reminds me. So another thing that we can do, because of the way the search boxes are implemented, you can also deeply link into this view and send links to other people. So if you wanted to send someone a link to a particular gene hit within this graph, right, or the same thing in our Manhattan plots, you can do that by just sending them a URL. And there are parts of the application where if we're reporting study results that happen to be significant in aggregate, and you click on one of the results, it'll zoom you right into that point here and highlight it for you. Okay, so the next section is the patients, and this is under the Explore tab. So there's a lot of different things you can do here. Here we're looking at just an individual patient. This particular patient, you can see they have this longitudinal data that's displayed at the top. So eGFR and UPCR, if you're not familiar, you probably aren't, I wasn't, are kidney health metrics. So these are displayed over time, and on the bottom axis you have like study days, so since the patient has come in. And just below that, those little green, blue, and pink tracks are medications that person has been prescribed over time. And so you can see how these things align with each other and how their kidneys are doing in relation to those things. And this is just useful for people who are doing research and they say, oh, this person has a certain genotype, or this cohort of patients have a certain genotype. Let's look at how they're responding to certain drugs, for example. So on the bottom, I guess we have a few tabs here. So there's clinical metrics that we report like full tables for. There's a whole lot of data in there. So there's sort of like a column selection for that if you want to show or hide particular information, if you're interested in, say, albumin over time. The medications timeline, and then there's also genotype information, which is basically about all the SNPs that we care about and how they're tracking in this particular patient, what their genotype is. Anything else I'm missing here?
Yeah, I would just note that there's a ton of data we're collecting about these patients. So everything from lab data, social history, demographic data, family histories, ICD codes, ESRD data, medication, comorbidities. I mean, ton and on and on. So lots of data, and a lot of these parameters we've exposed, so you can use those as filters to kind of build a cohort. And I think Adam's going to show that in a minute. But it's pretty powerful how we can filter on all these different parameters that we're interested in. For example, to look for fast progressors of kidney disease and different phenotypes that we might want to look at, and then can kind of dig into the genotypes that might be related to those patients.
Yeah, hopefully the next slide will actually show a little bit of that. So this third section is projects. So a project is basically, you can think of it as a filter set, which applies phenotypic and genotypic filters. So it could be clinical information about them, like how they've been diagnosed, whether they have type 2 diabetes or if they're a control in one of the studies. Demographics, such as their ethnicity, sex, and age. So you can filter on all these things, apply them, and then you can save those filters for later or mix and match them with other information. So if you're looking at a particular population and you want to see how a particular genotype affects that population, you can do that, and you can kind of come at it from the other angle. So here we're loading up. In the very beginning, you can see the project. So here we go. We're clicking on a project. It loads it up at the top. You can see the applied filters, the demographics tab. We're looking at overall for these patients, and then we're applying an ethnicity filter, and then looking at specifically the 600 and so many patients that have that particular set of criteria.
Yeah, the other thing I would point out is that we've put a lot of effort into thinking about how we can version all of our data over time, and so we expose that in the UI through projects so that you can view a project as the day and time when you save that project, or using the latest data, because we recognize that our data sets are going to continue to evolve and they're going to change, and we want to be able to go back to what the data was at any point in time and also look at the latest data. So it's added a lot of challenge from an engineering standpoint, but I think it's a pretty unique capability that we kind of baked in early on into the app.
So next up is a gene page. So this particular page, oops, sorry. Am I back? Nope. So this page, genes are sort of throughout the app. They're sprinkled pretty much everywhere, as you would probably expect. Here is our landing page for them. So this is where we can surface everything we know about that gene, whether it's been annotated in one way or another, by the target discovery group. We have additional resources linked on the right. And here we have patient variants. Let me see if I can click to start the animation. So these are counts. This is called a lollipop chart. And we sort of have an inverted version, so you can see cases versus controls in the top and bottom. And down below, you can see allele frequencies reported from Nomad and from Goldfinch. They can use this for QC, and they can also use this to determine sort of how rare certain variants are and whether they're potentially very deleterious or whatever. So there's another piece there which you can see the max MAF filter, and that's a minor allele frequency. So you can sort of say, I only want to look at really rare variants. I guess I'll add that the lollipops are colored by the variant impact. So high impact variants are sort of called out a bit. And then we also have this little flag, this concept of flagging, where there's certain variant effects that warrant being sort of called out. So protein, or let me think, protein truncating, I think is one, damaging missense. And basically, if something is high impact or has certain consequences, then we want to surface those variants at the top so researchers find them right away. Am I missing anything on here? And there's quite a few different features in this page.
Yeah, great.
So I have a little more detail on this one, actually. So here, these are study hits for a particular gene. And here we're surfacing, for every single study that's been uploaded, we're surfacing results that relate to that gene. So in the case of EQTLs and, for GWAS studies, we're looking at peak SNPs. So basically, if there's a peak SNP that falls within the gene body, we can report the value for that and sort of tell you what the value is. In the case of EQTLs, which are something quantitative trait loci.
Expression quantitative trait.
I don't know how I forgot that. Yeah, so they're associated with certain tissues, right? So we can surface the tissue and other information about that as well. And then you can click, as I mentioned, you can click on sort of the p-value or effect, and it'll bring you right into that particular SNP in that GWAS study, or gene expression studies. I think we have gene expression and RVAS reported up at the top.
Yeah, I think this is an awesome feature. So somebody on the team reads a paper and they say, "Oh, I wonder what we know about that gene," and so they can quickly just navigate to the gene, and now we can look at both public and private studies and see what are the effects of of that gene. What does our data look like? What does the public data look like? How can we compare those? And just get a really quick overall kind of view into that gene without having to go study by study and try to dig into that. So it's a huge time saver, and it just really optimizes our scientists' time so that they can do more with less.
Cool. So the last section is targets, which we talked about before, sort of coming from GWAS. So the target dashboard, which I'm going to wait until this cycles back. So here we're surfacing per gene study results that a particular person is interested in. So each user of the application can select a set of studies that they're interested in reporting and seeing. So if you're interested in a certain phenotype, or if you're interested in a certain clinical population, you can just report those studies and see how your favorite gene or your favorite locus is kind of tracking in those studies. So here we're clicking on a result, and it's sending us into the Manhattan plot for that. And you can see there's a SNP that's highlighted. It's zooming to that locus of interest. So I think at this point, my notes are a little screwed up here, but I think at this point, this view, we're sort of talking about additional features or additional data sets that we're surfacing through this app. So basically, this whole app isn't just like, oh, we're just showing genes over here and we're just showing targets over there. A lot of this stuff is kind of mixed together. So here you can see this sort of spark line at the bottom of the graph is recombination rate data. And you're able to click on individual points, or you can go to the variance table here and see variant effect data. And all those things are coming from different sources. And so we're kind of mixing them all together and joining that data, sometimes in the back end, sometimes in the client itself. So another piece of that is linkage disequilibrium data. So just the scale of this kind of boggles my mind, but The idea is for every variant in a GWAS study, and there are lots, you can sort of say, hey, how is this particular variant in linkage disequilibrium with any other given variant in the entire GWAS study? And so here we're reporting the R-squared values against that particular LD reference that they've set, and sort of lets them see whether or not that's a high or low value to identify other potential SNPs of interest. Adam, you could probably talk better to this point because I'm still pretty fresh on like learning about this stuff, but I do like making pretty graphs.
Yeah, I think this is great. So this is something brand new we're adding into the app. And I think the important part too is it's very context aware. So as you zoom out, some of these tracks disappear, we're displaying less. And then as you're zooming in, you're getting increasing levels of detail about the locus that you're looking at.
And just another sort of piece of data. We already talked about EQTLs as being something, so we're just looking at that. But we also have on the sidebar, as I mentioned on the gene page, a sort of list of different additional resources. It's tough. I should have kind of paused in the beginning here. But once this refreshes, you can see on the right side, there's additional resources which you can open, or you can just click this button to completely blow up your browser. And that's something that apparently researchers love to do, is just look up everything all at once.
I think the point is, you want to look at a lot of different evidence as you're evaluating a hit, if it could be a potential target that you want to investigate further. So, we're such a small team, we can't think about pulling all these data sets in and managing them in the app itself. And so, we wanted to provide convenience links to get to these other sites very quickly and efficiently, so that people don't have to go search through, multiple clicks to get to the gene that they're interested in at another site. So we've integrated all these in, as well as some internal references and data sets that we have where we don't have a study viewer built in the app yet for single cell, let's say, data, where we have some internal data sets related to this and we have a viewer for it, but it's not integrated into the app. So instead of spending a ton of engineering time trying to put all this together, we just provided links to make it really easy to go look at those other sites, whether they're internal or external, and pull that data together to kind of make an informed decision about what you're looking at.
Yeah, I think my favorite thing about this is it was a very small amount of effort to build a feature that I remember people being like, this is amazing. So yeah, user feedback is nice.
Yeah.
So at this point, I think this is all we have for demo of the application itself. We're about halfway through here, maybe a little more than halfway through. I just wanted to pause briefly and see if anyone had any questions about anything we've shown. I know there's kind of a lot of information and people are coming from different backgrounds, but if anyone has any questions, we'd be happy to answer them at this point, or you could ask later on. And at this point, I'm going to hope this chat feed thing works. Oh, and I was also hoping my battery wouldn't die.
They're going to be in the comment section now.
You want to be my my guy?
You got one. Someone's asking what kind of charting library you use for this?
Oh, that's a great question. What kind of charting library do we use? So we tried 3, 4. I've used Chart.js in the past, and that's what we ended up using. So we're using Chart.js. I use a wrapper around that called React Chart.js 2, and I wrote a wrapper around that. That doesn't have a name. So if you want to come up with a name for it, that'd be great. But yeah, that's as we said, this is built in React. So the thing with Chart.js and a lot of these libraries is they're written in an imperative style. And then when you try and wrap that in React, it's pretty, it can get pretty dicey. So it's important to sort of contain all the ugliness into one little place. And React Chart.js 2 does a really good job of that. And we just sort of expanded on it to allow like the animations and stuff that you're seeing there. It's annoyingly difficult to get those things to work just right. Any other questions?
I got another one for you. There are a lot of cool features. I imagine you had some pretty big technical challenges during this project. Can you share some of the biggest technical hurdles you overcame?
Technical hurdles. We're sort of going to talk about that a little bit later on Adam's part, I think. But I'm going to say, you know what? I mean, some of the technical hurdles that we thought would be very difficult turned out to not be very difficult. So one thing is the lollipop chart that we're showing here. There isn't really any state-of-the-art, there isn't any JavaScript library for doing this sort of thing. And so we had to completely write this by hand. And when we did the initial design sprint with Goldfinch, I looked at this and I said, that's probably going to be one of those things that takes, maybe a sprint or a month, who knows? And it actually came together really, really quickly. I think thinking about it more, GWAS is definitely one of the biggest challenges. It's where we have the vast majority of features, and it's one of the most used parts of the app. The challenge there is that the data is very, very large, and we're trying to represent a lot of it all at once. So one thing that we just ran into is we had two thresholds where you could go down to 5e to the -5 as sort of like a lower bound threshold for p-values. But we have some underpowered GWAS studies. Not all of them are, so we have to design this for the worst case, but at the same time, we have to be able to show really low or really high p-values, really low significance data. So the scientists can actually look at it and say, oh, is there actually like evidence underneath this peak? So they're looking for sort of like what's underneath the iceberg. And that terrified me, because when you look at this data, there is a very long tail. There's just a massive sea of points underneath all of that. And so to put that all on a graph or to try and render all that is obviously not possible. So we use a mixture of sort of downsampling and some magic on the API to kind of like abstract it away in just the right way so we can surface all that. I'd say that was definitely a big challenge. Yeah, I don't know. I think modeling, just real quick. So I think the thing that has been the most challenging is actually honestly just communication with the users to reach an understanding about how they're going to use this. We have a huge variety in like the ways that people want to use this application. Target discovery has been most recently driving how we think about building this. But in the past, it was more, we were thinking sort of a phenotype-genotype relation and from a clinical aspect. So those different conflicting, or sometimes conflicting, they're just very different ways to think about using an application. It's hard to say, this is the way the UX should be. This is how creating a locus of interest should be. So honestly, a lot of that is really just getting people sort of to the table to communicate what they want in an abstract sense, so you can kind of distill that to the UI.
Yeah, and we'll talk about product design in
the comment section.
Oh, geez.
So, and I know you guys have more to go. As makers, a big challenge is often ramping up on a project. With such a specific domain, I'm interested to hear how you were able to overcome some of those challenges.
Do we answer this later? So how we're able to overcome the challenges just ramping up?
Yeah, like ramping up, like, you're going to get some deep genomics there. So like, I imagine the question is like, what's it like to ramp up? How do you guys ramp up on that? And I know you had a unique advantage, so.
Yeah, I kind of cheated because I had studied biochem informatics in college. I was coming from a comp sci angle, so I by no means understood what EQTLs were when I came into this, or what LOF stands for, loss of function. Like there's all this stuff I had to learn out of the sort of like as I went, but I did have a decent foundation there. And I think I was able to share that with some of the other technical people on the team that are coming in from an engineering background and really haven't seen a lot of this. So in a way, there was some luck in overcoming some of these challenges and communication barriers. And I think that's certainly something that can help. I'm not sure if that answers your question, but hopefully it's close enough.
And last one, would love to hear about your collaboration with scientists to come up with solutions.
Collaborate. So I think that's the whole thing, really. And we're going to talk a little bit about that next, but it's really the whole thing here is just sort of lowering barriers between people with very different skill sets. And I think hopefully we'll answer that question in this next section.
Yeah, I think if I could jump in there too, I think, it's always been a challenge. We try to have meetings across different, with the various scientific disciplines. But I think one of the things that I have to say impressed me about the Rocket team is that, if they weren't getting the answers they wanted to questions that they needed answers to be able to develop features they were working on, because we're all local, it wasn't where they just sat there and waited for the next scheduled meeting. They literally just show up at the office and would walk around and sit down and talk to us. And, I think it give them a lot of credit where, they didn't just sit there and wait and say, oh, we didn't get the answer we wanted, so we didn't do what you asked for. So, they really drove it and pushed the conversations to get what they needed to really kind of integrate with our scientific teams.
Yeah, I think if you're aiming to build a project like this, you probably have to bake in a lot of padding for just communication and building understanding of both aspects in both directions. I mean, a lot of it is, from my perspective, it's just absorbing the biology, but I'm sure there's learning on the other side of just what the client is even capable of, what we can build, in a web browser. Certainly, a lot of times people are like, I just want to do this, and it's like, but is that exactly what you want to do, or do you want to solve a more sort of abstract problem that we can think of other ways to do it? All right, so we're going to sort of talk about, I think, more of this big picture stuff in this section. Nope, I'm not there. Here we go. So let's talk about the tech stack that we're using first of all. For our part, we have a React SPA, so it's a single-page app served directly from Amazon. It's coming from S3. This talks to an API built on a mostly serverless stack that's in AWS. Including Lambda, API Gateway, and Cognito for authentication. There are 3 distinct environments, which is pretty common for us, so dev, staging, and production. And all the code is deployed and tested using a CI pipeline that's built on GitLab. CI is continuous integration. So backing the API is an event-driven pipeline, which is pretty much the magic meat grinder. And that produces everything in the app that the app needs sort of on demand, so as a study gets uploaded, for example.
Yeah, I think the only thing I'd mention, so there's really only one persistent server in this entire stack, in which is one of our relational database servers. So the rest of the backend's really a carefully designed data lake that persists across numerous different data stores, including RDS Aurora and Athena. And so this is not only really operationally efficient, but very cost efficient, which was important to us. When someone uploads a new study from the app, the pipeline's kicked off, and then, as Adam mentioned, kind of the magic happens, and then that's how we kind of are able to distill a lot of these really large data sets down to something that can be served up through a client in a JavaScript app.
Yeah, I liked your description of sort of the pyramid approach.
Yeah, the data, we're going from petabytes of raw data to something that's really useful by our end users, and so the data gets processed and refined and distilled to a much smaller, kind of more manageable data set. So the data gets supplemented, enriched, and annotated with public data as well to try to give end users everything they need to better understand the genetic drivers of kidney disease and identify potential new drug targets that will help these patients that so desperately need new treatments.
Yeah, another piece of dealing with all this data is that even when it's scaled down significantly, kind of as we were talking about with GWAS studies, a lot of facets are still very difficult to interpret. So in order to enable target discovery, we also need to make it very easy for researchers to make quick, confident, and correct decisions about the data, not just decisions, but interpretations. So there are 3 ways that I see us doing this. One is through thoughtful design up front. So we did a design sprint, but we also are continually sort of challenging our assumptions about those things. Lots of iteration and tight iteration loops with users, and heavy collaboration with comp bio, statistical genetics, and bioinformatics at every level of the engineering. So from the data pipeline design to the interface design, there's so many things that could be missed there if you're not having those conversations with those people. So in the last point, I think those people mentioning that, it's all the breadth of knowledge that is on this team is probably the most impressive part of this whole project. It's not the thing itself, but it's the people that are backing it. And I feel like it's just this perfect balance between sort of breadth of understanding and overlapping of skills that we're able to do this. Can you speak to that a little bit?
Yeah, absolutely. So we worked hard to put together an integrated multidisciplinary team to work on this. It was really important to us that IT and software engineering, for example, weren't isolated from computational biology, but instead that they were working really closely together with a consolidated reporting structure. So we have a wide mix of skill sets, including comp bio, statistical genetics, data science, bioinformatics, software engineering, data engineering, and even more traditional IT and kind of cloud, architecture support. So when we were building out the team, it was really important to us that we found candidates for the various roles that were excited about not only going into depth on their kind of area of expertise, but also getting exposed to a wide breadth of opportunities and new technology. We're never going to hire someone that has a lot of experience doing a large-scale genome-wide association study on kidney disease, but we can hire people with the foundational skills that learn quickly, and that's really what we focused on. So the team works really well together. We do weekly code and technology reviews. For example, we use version control, we use containers, we have a good process for transitioning research prototypes into well-tested production-level pipelines.
Yeah, and just Easter egg on this slide. I mean, the whole bottom of this slide, and we do mean this when I say that these are sort of like the things that we sort of try to practice as much as we're preaching them here. But this is just the Agile manifesto. Like, it's the top 4 bullet points of Agile software design. It's not Scrum itself. It's just the sort of individuals and interactions over processes and tools, just pretty basic stuff. And I think it's working. Like, what we're seeing is it working really well with us in Goldfinch. So last thing we're going to talk about is sort of where this is all going, not just for the KGA and Goldfinch, but just biotech in general. Yeah, can you tell us a little bit about what we're sort of expecting to anticipate in the future?
Sure. I mean, there's a lot of technology behind the scenes supporting the KGA. So keeping track of tens of thousands of samples is no small feat, and we have a host of services and databases behind it. In addition, we're receiving clinical medication histories on these patients from numerous collaborators, each of whom stores this data in a different format and different system, and have a pretty wide range of technical capabilities as we interface with them. So we have both small and large scale analyses being done on these data, and some things that can be done on an individual server, for example, and then some things that require a large cluster of servers with a significant amount of RAM available. So in addition to compute and data infrastructure, I think the technology landscape in the experimental domain is constantly evolving as well. We have incorporated a mix of whole genome and whole exome short read sequencing data into the current KGA, but recognize that long read sequencing is steadily improving and becoming more commercially viable. Additionally, single cell sequencing has become much more cost effective recently, and the field of epigenetics continues to improve and expand. So all of these factors are really included for consideration as a plan is developed for a new study in the KGA. It's always important to balance speed, cost, and quality as they relate to technology, and finding the right balance is always going to be a challenge. One other aspect I want to mention of Goldfinch's discovery platform that we haven't really discussed yet is our ability to grow human kidney organoids. This platform gives us the ability to test our compounds in a human system. It presents an interesting challenge, though, as we start growing organoids from cell lines that are derived from patients in our KGA. So now we not only have primary patient data, but we also have derivative data and a hierarchical relationship between samples from a patient.
Yeah, the organoid stuff blows my mind. And just representing that stuff in the database is a whole other challenge, layered on top of enough challenges as it is. One thing that I often wonder about is, well, actually, before I get into that, what advice do you have for others in this industry that might be struggling with sort of the same problems that we're seeing here?
I think so data's being generated at an exponential rate, and that's not going to change anytime soon. There's a lot of challenges related to this, whether it's managing depth of a few types of data or a breadth of a lot of types of data in maybe smaller amounts. The problem of how to organize it and make sense of it is universal. The most critical aspect to consider initially is just how to organize it, and I think being thoughtful about how you might want to use it in the future. Even if you don't have the resources to build or buy systems to do that today, just organizing it in a thoughtful way can pay dividends down the road when you need to consume it, and you do have those resources to actually make sense out of the data.
So that actually dovetails into exactly what I was going to ask, which is like, where's the inflection points between in like data scale where it's no longer simple enough to just cram it all on the database and model it relationally?
Yeah, I mean, there's some strict limitations in terms of how much data you can reasonably put into a relational database before it starts falling over. There's ways to extend this, such as sharding, but there's also some fantastic NoSQL solutions that exist today. So, those make it a lot easier to just dump data into an S3 bucket, for example, and query it. And there's a variety of file and data formats that you can put it in. So designing a fully normalized relational database requires really a considerable amount of effort and upkeep. So these NoSQL solutions can provide really good query performance. But it's challenging when you think about the relational database because you want that query performance, but you have to consider scalability as well. So it's always good to think about not only kind of the short-term need, but also where you're going to be in 6 months or 12 months and down the road.
Yeah, definitely not a job for inexperienced people.
No, but I think at the same time, AWS and other cloud providers are adding a lot of service offerings to make it easier and easier. Every industry is experiencing the data deluge, and there's lots of architectural options for how to handle it. When we were looking at solutions for the data lake, for example, we looked at several columnar data warehousing solutions, and these can be great tools, but are not always the right tool for the job. So it comes back again to balancing cost, performance, and the resources that you have available to work on this.
Excuse me. So let's shift our focus a little bit from the data and the tech in general to more focused on engineering and product design. So this is a bit of a loaded question since it's what we do, but what role does engineering and product design play in advancing the Goldfinch mission, and how do you see that evolving in the future?
Our platform is built around the patient. I think a lot of biotech and pharma companies say something to that effect, but our whole pipeline starts with collecting data from patients that have kidney disease. And so because of this, it's increasingly important that we have a strong engineering team helping to build out the infrastructure to manage, analyze, and share all the data and insights that drive our target identification pipeline. Sitting on top of this is product design, of course. So, often in scientific domains, this is completely overlooked. It's really important to us that the product we designed was a professional software application and that it looked that way. So thinking through the workflows that we wanted to optimize, designing the interfaces, studying user interaction, these were all important aspects of what we were building. We were also discussing new ways that we can do a better job of interacting with the patient community and getting them engaged with our Kidney Genome Atlas. So as we continue to think about this, having a software product that's really end user focused will be absolutely critical.
So you're sort of touching on this, and the slides, the goofy slides here touch on it as well. But what are your thoughts on the differences between delivering something that's just an internal tool versus something that is for B2B or like direct to consumer?
Yeah, initially, our platform was designed to be an internal tool for use at Goldfinch. I think a lot of scientific applications, never expand beyond that scope. But as the KGA has continued to evolve and expand, we started to bring in external partners, and we also took into consideration how we could use the application to share the data we're generating with our partners. So thinking about how the KGA might continue to evolve, we're just starting to think about new collaboration features, such as allowing contributors to see some of the data they contributed to the KGA, or patients who are individual contributors. So the scopes really continue to evolve with the scope of the KGA to support the business's needs. And so as the scope has expanded, there's lots of technical considerations that accompany this, such as thinking about permissions and roles and increased security, and then data segregation so that we're not incorrectly sharing data with people who contractually aren't allowed to see it.
Yeah, the first thing I think of when I think of internal versus consumer facing is that there's a lot more time spent with designers. And it's especially true, for consumer facing versus B2B, but it's true for both. But stakeholders often want to get it just right, and it's really difficult to balance that with being agile and sort of putting out an MVP and iterating quickly on that and getting to sort of the meat of the problem, and serving the real world problems that you're sort of intending to serve. I'm wondering how you would describe sort of your approach to building this platform.
Yeah, I think the approach and design of the application has been very iterative. When we first started out, we had a really loose idea of one workflow we wanted to design around, and some of the data that we would have to surface to enable that workflow. Over time, the scope's dramatically expanded. Each new user that we get has kind of suggestions, ideas, preferences. We're a very small team, so we don't have a dedicated product manager, and we prefer to be really lean and agile so we can respond to requests and shift priorities quickly.
So this actually was a question that somebody had asked sort of midway through, but what were the biggest challenges doing this?
Yeah, I think one of the biggest challenges that comes to mind is obviously just the amount of data that we're trying to surface, and how do you summarize and display that in a meaningful way? So it was a significant design challenge initially, and then it became an engineering challenge with how to implement a responsive, scalable JavaScript application. The other significant challenge that I want to mention is that we're building an application as we're building the rest of the KGA. So we don't have this polished specification document where we can just throw it over the wall to engineering and say, this is exactly what we want to build. So, we're defining this on a weekly basis. And as we produce new data sets and we add new capabilities to our analytical pipelines, then we have to quickly kind of adapt and figure out how we're going to incorporate these into the app.
So last question, what have you learned in the process? What are your sort of key takeaways that you'd like to share with people?
Yeah, good question. So, I think it's critically important to have a key set of stakeholders that are involved in the project from day one. And these people need to be really strong advocates as end users of the app to the business of how critically important a project like this is and what business value it adds. So, developing software, it's not cheap, it's not easy. And unfortunately, finance kind of sees dollars and cents in this scenario. So you need to have champions who can translate that cost into the business value that it provides. So it's always challenging to decide on building software versus buying it. I think you can spend an eternity evaluating the landscape of what's out there, looking at different vendors, building those vendor relationships. And this can be really time consuming to build and manage those relationships. So it's important that whichever path you choose, I think that the business is really committed to it long term and understands not only the initial investment, but the recurring costs and what that looks like multiple years from now.
Awesome. Well, Adam, thank you so much for your time and thoughts on all this stuff. That concludes sort of the canned portion of this presentation. We'll take some more questions if anyone has them. I think I'm going to need to sort of show this bit because I can't see. Okay, here we go. Oh, all right. Do we have any questions? Let me go up a slide here. I think I'm looking in the wrong place. Hey, Jesse, where are you? Save me. No, this is on YouTube, but I don't know what a link is.
Not yet. So
nothing yet. Okay, great. Thanks, man. I thought the place I'd see them is here, but all I'm getting is trolled by people I work with.
I'm just getting trolled by Matt Jacob. So,
I saw that. Classic. Well, we got some good questions midway through, so I don't know. I'm happy with that.
You guys could go back, like at the very start of it. Is there any like big fundamental things you guys would do differently at this point?
Did you hear that question, Adam? So the question was, if we could go back to the very beginning.
Get in your DeLorean, go back to 2019, maybe 2018. Like, what would you do differently, if anything?
Yeah.
Yeah, good question. I think thinking back in with our perfect hindsight, I wish that the team we have today is what we started the project with, and of course, you're never going to have that luxury. But it's, I think one of the challenges, and when I think about some of the features we built in, the order of things that we did were based on what we knew at the time. But I wish that we maybe had been a little further along in terms of the project to really understand more clearly the specific workflows that we wanted to optimize. I think that was one of the challenges. Was we had kind of a loose idea initially of one of the workflows, and they've kind of evolved over time as we've added people to the company, as we've added kind of technical capabilities as well. So I think that just trying to kind of get a clear sense of upfront kind of what our requirements were as much as possible would have really increased our efficiency a bit, especially in the beginning of the project.
I got no comments. I don't have a DeLorean. You got more? Oh, so question is, what are the next steps in the collaboration together, and what's on the horizon? I'm gonna leave that for you.
I think, from my perspective, we're constantly looking at adding new data and just what we can do to support our scientific teams, and so looking at how we can expand across, make the app more useful and applicable to other parts of our R&D organization, and then also, as we're generating kind of new data sets, thinking about how we can incorporate those with what we've already built, so that we can just continue to give people, a really good picture of what the data in the KGA is saying about various, genes and variants that we're finding as time goes on.
Cool. All right. Well, if there aren't any other questions, I think we could call it a wrap. Is there another one? What do we got? from an engineering perspective, what scares the hell out of you? I mean, the data, I think, is definitely the easy answer here. As terrifying as the data is, and as sort of gigantic and difficult to manage as it is, from my perspective, it actually hasn't been that bad. And most of it has really just been fun because they're new, interesting problems to solve. The reason it hasn't been that bad is because we have data scientists to lean on, and we have the comp biologists and all these people that are able to sort of make sense of the things and provide sort of the right abstraction to build that pyramid up to where I am, where this data still looks gigantic, but it's not, petabytes with a capital P. So that, honestly, as scary as it was, the thing that should have scared me more was understanding the science and being ready to communicate as much as is necessary with sort of the people that are consuming this. You have to have a really low barrier for asking questions about this stuff, because I think I came in and I'm like, I study bioinformatics. I'm going to get the basics. Like, we're going to, bang this thing out, no problem. And every other acronym was a completely new thing. And every concept I had to sit down with their lead scientist and go, can you explain this to me? And we spent a lot of time drawing on whiteboards and whatnot. And that's tough, because I think a lot of clients don't think about that particular aspect. That, to Adam's point, he said, the biggest piece of advice that he would give, I think you said, is to have those key stakeholders involved. So when we do a design sprint and we're like, oh, let's, we're going to get these stakeholders in the room and get them to work on stuff. That's the whole idea, is to sort of get the right people to distill the right information and, kind of set things off in the right direction.
Yeah, I think the other challenging part is, again, because we're not meeting every week and saying, here's exactly the features I want you to go build. Here's what you need to add to the app. A lot of it is Adam talking to end users and saying, well, I kind of get the sense that, this would help them out, and so I think we should go do X, Y, and Z. So, Adam and team, at Rocket have done a great job really interfacing with those people and starting to kind of, try to think like them, think about what they need, even though they're not asking for it. And I think that's a big part of the challenge.
How has the application changed how teams interact at Goldfinch? That's a good question. It's also coming from a troll, so if you want to ignore it. Matt Daigle. Got to get your digs in when you can. I'm going to leave that one to you. How do you think this is changing the way teams interact? I certainly know how we're hoping.
Yeah, I know. I mean, the traditional model in a situation like this is that, the computational team's generating a lot of this data. So when you want to go look up information about a gene or a variant, you go email the computational team and say, what does our data say about this? And you wait a few days and you get a slide deck back. And, that's one of the things that we were really trying to get out of the habit of and kind of break the mold with this app, where, now people are empowered to go look that up themselves and make their own decisions. And it doesn't all have to funnel through the computational team, and they can really sit down and focus on how they can add additional value beyond that, and coming up with new analyses, new data sets, new processes that we're working on, as opposed to just creating slides all day. So, I think we're getting there. I think that the app has been really a great enabler, towards working towards that. And so I think that's a really important aspect of how we're kind of changing the working style.
Cool. Seeing some of the comments. All right. Let's call it there. I think I'm done. And I'm sure the folks in Europe are like, hey, let's call it a day. Let's go get a beer. So yeah, thanks. Thank you everyone who stuck it through this long. Appreciate it. Thank you, Adam, so much for doing this with me. Yeah, and sorry, we both have the same name, to people who are confused as to who's who.
That made it easy.
Yeah, it's not too bad. All right. Well, I'm going to hang around until everyone is gone. That is what I was told to do. And I'm going to do it. And I can't tell how many people are here. All right. I'm going to go to slide mode.